Hello everyone and welcome to this class. In this class we are going to study about endospore formation. Okay. So we will first start with the conditions. Okay. So there are several conditions in endospore formation. So basically there are bacterium like bacillus and clostridium which when they are under stress conditions which can be heat, desiccation, total dehydration or no nutrients. So under these conditions you can say that a bacterium, as I was saying, let us say we are taking bacillus here. A bacillus will feel stress, and in that stress what will happen is that, let us say this is the bacillus bacterium. Okay, so and here comes heat. When this heat will fall on the bacterium, then a certain signaling transduction pathway will get activated, and due to that activation a certain factor which is SPO0A, it will get activated and phosphorylated. Okay, so now what will happen is that when this factor will get phosphorylated, then certain genes which are responsible for sporulation or endospore formation will get activated. As this SPO0A, it masks that particular gene. It masks this gene right here. Let us say this is the SPO. It will mask this. But when it gets phosphorylated, it will get removed from this particular gene, and after that it will act as a DNA replication inducer. Okay, so then this will replicate, and what will happen is then this bacteria will start the DNA replication. DNA content will double, and also the cell will enlarge itself. This is our cell. It will enlarge across the axis. Okay, so let us say this DNA has replicated right now. So what will happen after that? It will create a particular septum here. Okay, this is septum, and when it will create the septum, as you can see, this septum is not symmetrical. Okay, it is a asymmetrical separation of this bacterial, you can say, whole cell. Okay, so after this what will happen is that this septum will, it will grow and will divide these two bacteria into two parts. Okay, so one genetic material will be here, another will be here. This part here is called forspore. Okay, here forspore, and this here is mother cell. Basically they are two, but they are one in itself. Okay, so what will happen after this is that this mother cell will go and it will try to eat up this forspore, eat up this part, this particular part, this here, so that this part will get engulfed into mother cell itself. Okay, so it will happen like this. Okay, I'm really sorry. See here, this will somehow get engulfed into the mother cell, and this is also peptidoglycan, and this right here is also peptidoglycan. So you can say when it will get wholly engulfed, then there will be two peptidoglycan layer for this one endospore, and this is the whole mother cell. Okay, so as I was saying that two layers will be formed here, so I'm going to zoom it a little. Okay, so let us say this is one layer of peptidoglycan. Okay, and let us say this is also peptidoglycan and with some another layer. Now in between these two layers, a third layer is going to get formed here, which is of cortex. Okay, so this cortex layer is going to form in between these already existing first layer and second layer of peptidoglycan. Okay, so after this what happens is that when these layers are formed, including the cortex layer also, so another layer, not one or two but several layers, are going to cover this particular endospore cell inside the whole cell. Okay, so you can say it is going to get very rigid so that it can resist, or you can say save the nucleic acid from another stresses. So after this what happens is that dipicolinic acid plus some molecules of calcium ions, they get deposited inside the nucleic acid of this endospore. Okay, in the nucleic acid of endospore. Okay, they will get deposited into the nucleic acid of endospore and they will stabilize this nucleic acid. And after this stabilization, you can say that overall coating and cortex formation and stabilization of nucleic acid when happens, then this outer cell, the actual mother cell, will degrade due to the stress that is in the environment. Okay, so mother cell degrade, and when this mother cell degrade, then what is left? We have only endospore left. This is how endospore is formed. Okay, now before closing this lecture, you should know what the properties of endospore are. Okay, so the first property will going to be, it is very resistant to heat, resistant to you can say moisture damage, resistant to UV radiation, and as the endospore is getting formed, this whole endospore is going to go into a dormant state. So basically it will just sleep inside the endospore. I mean, main nucleic acid will sleep inside the endospore. Okay, so it is dormancy. Okay, means it will not carry any metabolic activities or any other activities for which it requires energy, and it will only form the vegetative cell. Like if this is endospore, and let us say that this mother cell has degraded. Okay, but this endospore can again form vegetative cell. And when will it form vegetative cell? It will form vegetative cell only when the conditions are favorable. Okay, heat resistant, moisture resistant, UV radiation, dormancy, and also osmotic / pH / other changes resistant, high survival rate. And the special thing about this endospore is that it contains only, you can say, 0.1 to 1% of water inside the endospore cell. Okay, that's it for this video. Thank you very much and see you soon.